By now, we have finished basic consideration of the heart functioning and assessment of the heart function using various methods of investigation. And now it is time to learn how the heart function is adjusted and regulated in order to maintain necessary level of blood pressure and finally to meet the body oxygen requirements. So this is a regulation of the heart. And we begin with classification of regulation mechanisms. Classification is very easy and it divides into big two groups, which is intracardial inside, intrinsic built-in mechanisms of regulation, the characteristic for the heart itself, and extracardial mechanisms. As for extracardial mechanisms, they in turn can be divided into two groups, which are, as usual, types of regulation division, which include neural and humoral mechanisms. Neural by nervous system, by nerves, and humoral mechanism by something that goes through fluid, humor, which means by hormones, by biologically active substances that can act through receptors. First, we consider the intracardial mechanisms of the heart regulation. They also can be subdivided. They all are based on the intracellular mechanisms and also they can be called myogenic, produced by muscle tissue. These changes may influence the heart function. And they are divided into hetero and homoeometric. The terms hetero and homoeo, the roots of words mean different. In this case, heterometric, it's a different length. If length changes, it may produce changes in the heart functioning. We already studied this law, but we'll come back again to this. And homoeometric with the root homoeo, which means the same, similar, unchanged. So homoeometric without change of length. And while heterometric operates through stretching of the fibers so that to influence the heart force of contraction. Homoeometric mechanisms do not require change of length, but their mechanisms are based on the accumulation of calcium ions in the myocardial cells, in contractile cells of the heart. And this occurs when the heart rate increases. And we'll consider this in details next slides. First, heterometric. Again, heterometric, different lengths, where changing in length produces changes in the force of the contraction of the heart. And generally, with stretching, the heart produces the stronger contraction further. And until the moment we reach this optimal stretching, this force increases and increases. Practically, it occurs due to a higher degree of the overlapping, so that number of possible sites for actin myosin filaments binding increases. And tension becomes higher. Tension here means practically the force of contraction. Something like this we have already studied, and this is characteristic not only for the heart but also for the skeletal muscle. And when this optimal stretching maximal necessary for produce maximal response is reached, further stretching will decrease and decrease progressively the tension developed by the heart contractions. To review this, let's recall length-tension relationship for the skeletal muscle. Practically, mechanism is the same. Optimal stretching permits maximal binding of cross bridges for maximal muscle tension. Of course, with words it's less easy to understand, but first you study this, and second we are going to see the pictures. And pictures explain this. Let's recall the structure of the sarcomere of the muscle. Here you can see unstretched muscle. Usually schematic pictures of the sarcomere do not show this overlapping of actin filaments in the middle of sarcomere, but really it can happen when muscle is entirely not stretched. And you see, this big portion of the actin filament is not used for contraction. It's not used by myosin cross bridges or heads. And all heads participate in contraction in this case, but this long portion of the actin filament in the middle is not used. And the distance for maximal contraction, look at this, is very short. Just a little movement of actin filaments to the center of sarcomere, and ends of myosin filaments will be exactly near the Z line, the discs, and further contraction is not possible. So there is no great space for movement, and contraction becomes weak in an unstretched muscle. From this position, if we start to stretch progressively with an increase of stretching, the force developed becomes higher and higher until we reach the optimum considered to be reached in moderately stretching of muscle. And you see here exactly this is optimal because the ends of actin filaments are located just nearby the first, the most medially located, the heads of myosin. So that all these heads present in the myosin filament still participate in contraction, and there is no any unused portion of the actin filaments. And if we look more distantly, you can see maximal distance now for between Z lines of the discs and myosin filament ends. And this position, until this stretching, we can have higher and higher and higher force, and now we reach this best situation with optimal. Here, with this stretching, stimulation of the skeletal muscle produces the strongest contraction. Any further stretching will produce decrease of the participating cross bridges number in the myosin filament. These heads of myosin will, not all of them will participate, and clearly the force will become decreased. So between left side and right side, anything makes higher and higher and higher contraction force, and the right side, the picture shows maximal force. Further stretching will start to decrease force, and here you can observe the overstretched muscle. You see this is excessive stretching, overstretching. Actually, it's hard to achieve in a real body because the points of attachment of the muscle is to bones, and bones are connected by joints, and their distance cannot be so much increased like here in the laboratory conditions. But you may observe in the picture of the sarcomere that there will be very, very little force produced, very low tension, because overlapping is practically absent, and number of participating cross bridges or heads of myosin participating in interaction with actin filaments becomes very little, just as compared to all the heads which participate in the best situation, and now just a little part of them. So it doesn't matter that distance to Z lines is high, but it doesn't help. The interaction is very little and contraction becomes extremely weak. And exactly this mechanism is very important for the heart, more than for the skeletal muscle, because again, skeletal muscle hardly can be stretched so much. But the heart muscle has no bone attachment, there is no limitation for stretching. Of course, the limit exists, but the blood that can fill the heart can stretch it more and more, and it can stretch so much that it produces already decrease of contraction. And in case of the dilation of the chambers of the heart, the contractions become extremely weak, and this is cardiac failure. So, and this is basic law of the heart, Frank-Starling law, that is based exactly on the considered laws. Length-tangent relationship that we consider on the example of the skeletal muscle operates in the heart muscle, and this is basis for the classical Frank-Starling law of the heart, which we have already considered in previous lecture, but let's recall again. The increase in cardiac fiber length-tangent relationship first, which really is produced by an increase in the end diastolic volume, but now for simplicity we can omit this. It results in the increase in tension or force of the heart contraction. So let's move back. End diastolic volume is an important factor that stretches the heart fibers, and this stretching until a certain degree produces higher and higher force of contraction. And maximum is considered to be achieved at the moment when length increases about up to 50% from initial length, so 1.5. it increases and then it starts to decrease force of contraction. It's around 50. In some sources you may find about 45%, but approximately up to 1.5 from initial length. Stretching produces reaching maximal force, and then further stretching produces decrease in force of contraction. This figure shows this. Values are very approximate here, but generally you may observe that end diastolic volume elevation increase produces progressively higher and higher stroke volume, which depends on the force, depends on the tension developed by heart muscle during contraction. But finally you see that until stretching is more or less moderate, dependence is linear, it's a straight relationship. But then stretching, excessive stretching, produces decrease of tension. Let's recall the normal values, which already we mentioned many times, but again, stroke volume, average volume is 70 milliliters with a range between 55 to 90 approximately milliliters. Then end diastolic volume, the full volume, is about 120, 130 milliliters on average, and range is from 90 to 140 milliliters for resting state. And ejection fraction, very important index that is based on these two, and stroke volume and end diastolic volume. As fraction name, it shows that it's part of. Which part of the end diastolic volume is made by stroke volume or systolic. So systolic volume divided by end diastolic volume and taken in, multiplied by 100%, so we take in percentage. Generally makes about 55 to 60%. It's a regular level for resting state. But range includes rounded value 50 to 75. So 50 is lowest, still normal, not the best, but still normal value of the ejection fraction. Anything less will become already decrease. And the greater decrease, the worse is the heart function, heart contractility. And at rest, 75 is not often present, but if activity increases, physical activity, the value of ejection fraction increases from 75 higher and higher progressively and can reach even, according to different data, even 95%, but never 100%. So the heart never squeezes all the drops out of the ventricles. Every time there is an end systolic volume, which is difference between end diastolic and stroke volume. And look here, the figure shows more or less correct values for classical end diastolic and stroke volume. And what is the significance of the Frank-Starling law? You see, it ensures the equality of cardiac output to coming volume of blood, venous return. Look how we can prove it. The end diastolic volume that determines the force of contractions of the heart depends on how much blood comes back from veins into the heart. So it's venous return. And therefore, if force of heart contraction depends on the end diastolic volume, we can go further and say that venous return determines finally the force and generally cardiac output. So stroke volume and cardiac output correlate directly with end diastolic volume and finally with venous return. And this is very important, that Frank-Starling law provides that the volume that heart moves to the systemic circulation, for example, in general, ejects and systole, equals the amount of blood that comes to the heart, which it receives. So there is equality also between income of blood and output of blood. So it provides non-stop movement of blood without excessive accumulation before the heart, before the right ventricle or before the left ventricle. So it contributes to make the work exactly as necessary. As much blood comes to the heart, the same amount should be pumped further and further. And so it continuously provides blood movement without accumulation of excessive parts. In a steady state, therefore, the cardiac output equals venous return. And all this is due to myogenic mechanisms or intercardial mechanisms of the heart, heterometric type. Now, let's look at the homeometric mechanisms of the heart regulation. And the best example is the positive staircase effect. Let's see what is this. First, let's recall homeometric means the same length without change of length, but something different is changed. When heart rate increases, from certain level of heart rate, the tension developed with each contraction becomes progressively higher and higher, of course, until certain reasonable limit. And this increase goes in a stepwise fashion. That's why it's called staircase effect. According to the picture, you will see it easily. Look, if we record the mechanical contractions of the heart, like usually it can be done on the isolated frog's heart. It's a mechanical record, mechanogram of the heart. You see, the contractions are reflected by ascending part line, then descending line. All this is contraction, and then pause between contractions will be lower position of line. And each upward loop responds to one systole, one contraction. Frequency increases, so we stimulate higher and higher. It shows how heart rate increase influences the force. And look, it becomes higher, higher and higher force of contraction. Of course, perhaps not as high as this picture shows, just it demonstrates well that it greatly increases, maybe not as much as 3 times maybe. So amplitude increases because to the horizontal axis we have gradual increase in frequency. How it can be explained? It's rather easy to explain. With each next impulse that comes earlier and earlier, the interval of time between contractions decreases. What should happen during this interval naturally? For each contraction of the heart, the calcium concentration in the cytoplasm must be increased. If you remember, the sources of calcium for the cardiac muscle cells, we have 2 sources. One source is extracellular fluid, and from extracellular fluid, calcium enters through channels of calcium in the T tubules. And also we use the sarcoplasmic reticulum. It's less developed than in skeletal muscle, but still it's present and releases calcium. And this is a necessary source to increase calcium concentration for contraction. But after each contraction, we should have diastolic relaxation. For the relaxation, each time we need to decrease this concentration of calcium. And here pump operates to decrease calcium concentration, to move calcium into sarcoplasmic reticulum. And also there are exchangers in the outer membrane of cells which remove calcium in change with sodium. But all these ways must decrease calcium concentration, and only then relaxation can happen. And with each progressive increase of contraction, we have less and less time to decrease calcium concentration. And so calcium slightly becomes not fully removed, and concentration is slightly higher than before. But next impulse comes even earlier, and so it goes each time earlier, and more calcium is still present inside the cytoplasm. Therefore, calcium provides stronger contraction. If you remember, it's true for any muscle, skeletal muscle, cardiac muscle, smooth muscle. Higher concentration of calcium in the cytoplasm provides greater force of contraction. So, shortening of the time intervals between each contraction does not allow incomplete calcium pumping back to reticulum, and calcium concentration increases with each next contraction, thus providing the increase in tension. And look at this. Exactly, it resembles the staircase. So that's we call it Bowditch staircase. The Bowditch was the first who described this phenomenon, and so it's named staircase according to its view with stepwise fashion. Then we can move to extracardial. Before we considered intracardial mechanisms which operate even in the isolated heart, like frog's heart, for example. The extracardial mechanisms for isolated heart of the frog we cannot use all of them because nerves are cut if we if we isolate the heart from the body. So nervous mechanism will not operate. But humoral will operate because if we add adrenaline or acetylcholine or something else into fluid that supplies the heart, so receptors are still preserved in the cell membranes. Therefore, response to these biologically active substances or hormones or neurotransmitters will happen as usual, will occur as before. So extracardial only humoral mechanisms still can be used for isolated heart. But let's come back to normal heart, which uses both neural and humoral. And first we must consider neural mechanisms, which are basis for further mechanisms of the humoral influences, because nerves release transmitters and the transmitters bind to receptors. And finally, humoral substances bind to the same receptors. Therefore, if we consider the neural mechanisms, they will be a basis for understanding of humoral mechanism influences. And neural mechanisms, the heart is innervated, as you remember, by autonomic nervous system. I hope autonomic nervous system is remembered well, and for the heart it's easy. Two opposite influences from sympathetic and parasympathetic system divisions. Parasympathetic part is the number 1 nerve for parasympathetic, nervus vagus, number 10. And the sympathetic part is represented by a nerve that is called sympathetic nerve simply, or according to Latin, nervus sympathicus. And stimulation of these nerves provides opposite effects. And it's hard to make a mistake when you remember that sympathetic system provides higher level of activity of the body. And clearly, exactly this nerve should stimulate the heart and produce higher force and higher rate of contractions. Why vagus nerve produces decrease in both these indices, as generally it contributes to the resting state with less activity, with closer to minimal activity. Generally, all vagus effects are inhibitory, while all the sympathetic effects are stimulatory. This is background which is present in everyone, I hope. And now we can recall a little bit of anatomy. Also, it's something that you studied maybe not once. As for parasympathetic part, which is nerve vagus. Vagus nerve has location of its nuclei in medulla. Large nuclei located in medulla contain the first neurons. You remember that generally the whole autonomic system has two efferent neurons. Usually first one within the central nervous system and second one in the ganglia. So first neurons exactly in the nuclei of nervus vagus in medulla. Then second neurons. If you remember, parasympathetic system has location of ganglia in the walls of organs mostly, which is called intramural, inside the walls. So the preganglionic fibers go directly to the innervated organ, long preganglionic fibers, and reach the second neurons just inside the heart in this case, cardiac intramural ganglia. And therefore their postganglionic fibers are short because they are already in the innervated organ, in the target organ. Neurotransmitter which is released from ends of the nervus vagus is acetylcholine. Actually, it's released even from preganglionic fibers as well. But final is acetylcholine, and it binds to the muscarinic cholinergic receptors. Only peripheral receptors are muscarinic, while in ganglia receptors are nicotinic. But we are interested now in the final effect, which is done through muscarinic receptors. And what is innervated in the heart? Actually, not the whole heart has innervation by fibers of nervus vagus. Right nervus vagus, we have both, two of them. Right vagus innervates the right atrium and the first node, which is located in the right atrium, sinus node. While left one innervates the left atrium and next node, atrioventricular node. And generally, you see it's all. Only atria are innervated by vagus and conductive system. So you can notice that ventricles do not have vagus innervation. Then sympathetic part. And this sympathetic nerve, or nervus sympathicus from Latin. First neurons are located. I hope you remember that first neurons of the sympathetic system are located only in medulla, or only in the spinal cord, I want to say, not in medulla, not above, only in the spinal cord. But here is the upper portion of the spinal cord, lateral horns of the thoracic first three segments, thoracic 1 till 3 segments. And if you remember, it's lateral horns, not posterior, not anterior. Then, second neurons, where are located second neurons? In the sympathetic system, preganglionic fibers are much shorter because ganglia are located very close to the spinal cord. And really this is cervical sympathetic ganglia, or ganglion stellatum, star ganglia. And transmitter is the norepinephrine. I hope it's clear that parasympathetic transmitter is acetylcholine. And in all preganglionic fibers we have again acetylcholine, and the only one difference is made by postganglionic fibers of the sympathetic system. This is norepinephrine. And this substance, transmitter, binds to receptors of different types, but in the heart this is beta-1 adrenergic receptor. All beta receptors, I hope you remember, operate through cyclic AMP. Adenylyl cyclase pathway is activated and cyclic AMP is produced. Cyclic AMP further activates the protein kinase A, which phosphorylates numerous substrate proteins. And innervation here goes for all structures of the heart. But to be exact, which structures are included? The conductive system, which means all nodes and fibers, and atria, and, which makes difference from parasympathetic system, also ventricles. Ventricles are densely innervated by sympathetic system. And this is the reason why sympathetic system can so much influence the force of contraction, actually increase. But even absence of fibers of nervus vagus in the heart and ventricles does not make problem for this nerve to decrease force of contraction even in ventricles. The heart efferent innervation. So let's recall now by picture that all that was listed before. First, what is innervated? Sinoatrial node and AV node, both. Of course, AV node cannot be seen on the surface, just this picture schematically shows it. It's hidden inside. And from medulla, from nervus vagus nuclei, fibers of vagus itself pass exactly to these two nodes. But if you remember, sinus node for innervated by right vagus and AV node by the left one. And also atria are innervated by vagus fibers. Then, as for sympathetic innervation, it comes from the spinal cord, thoracic segments 1, 2, and 3. And it comes to ganglion. The medulla itself is not shown with first neurons, but second neurons are installed in ganglion. And from this ganglion, further fibers, postganglionic fibers, come to the same nodes, which are innervated by vagus as well, and also to cells and to the atria, and to conductive system in ventricles, and to ventricles muscle tissue. So ventricles makes difference again. So this is innervation, efferent innervation for the heart. And so if we need to increase heart force and rate of heart contractions, we may stimulate the sympathetic nerves. For decrease, we can produce activation of vagus nerve. Actually, at rest, when we don't perform physical activity, we don't need great activity of the heart, and in resting state, heart is controlled predominantly by nervus vagus. Its influence is clear, present at rest, while sympathetic system influence at rest is negligible. And vagus constantly sends impulses to the heart. So, to say the truth, real frequency of the sinus node is not between 60 to 90, with medial level about 75. Really, it's closer to 100 if we exclude vagus. But we study values for natural situation when heart is under constant influence of vagus nerve. Really, sinus node could generate it faster, and it happens when vagus influence decreases. Main factors for the heart function. These are rate of contractions, heart rate, and the force of contractions or tension developed during contraction. These are main factors. Additional factors are excitability and conductivity. These last two factors, actually, when they increase or decrease, they make contribution to a corresponding increase or decrease of the main factors, rate and force. Basic factors are contributed by higher or less excitability, higher or less conductivity. But total number of factors really is not 2, but 4, with main 2 and the others additional. And let's observe first influence on the rate of contractions. It's a different mechanism for, not entirely different, but at least different target cells for regulation of the rate and for regulation of the force of contractions. To regulate the heart rate, remember which cells we should influence. The cells that drive the heart, that control the heart function, which is called pacemaker, who makes the pace of the heart, who drives the heart to be contracted or not contracted. Because heart muscle cells maintain their resting potential constant without changes if there is no stimulation. And stimulation comes from the conductive system cells and, of course, firstly from the sinus node cells. Let's recall, this is the cells of the sinus node that regularly generate impulse automatically due to spontaneous depolarization. This is from -60 to -40, this is slow diastolic depolarization phase. It occurs slowly, but it regularly brings the potential to the threshold level. And this threshold is reached at -40. And when threshold is reached, every excitable cell will produce, in response, action potential. But so this depolarization phase starts from this moment, and then repolarization, but not many cells are able to do automatical reaching to that threshold, which is normal for sinus node cells and generally for conductive system cells. To understand clearly how we influence the heart rate, we should recall in details the mechanism of the slow diastolic depolarization, how it occurs. Depolarization occurs first. Now let's consider the schematic representation of the cell of the sinus node. For all the excitable cells, usually the resting potential is created by constant little loss of potassium because of high permeability for these ions, and difference of concentration is present and directed outside. And due to this, we have negative charge on the inner side of the membrane, while potassium creates positive charge on the outer side. And so this is a reason of the resting membrane potential. But for these cells, this resting membrane potential is not maintained. Cells do not need any stimulating factors, and they constantly lose this -60 and make less and less minus. This is unstable resting potential. And why it happens? Let's observe. First figure, -60 is this resting potential, which really is only points reached, and then again the potential comes upward and upward. This is the threshold level, -40, and slow diastolic depolarization. This phase occurs due to changes of permeability for all 3 ions that participate in this process. The letter P stands here for permeability. And permeability actually reflects the number of channels, open channels available for ion. And so if permeability increases, it means that more and more channels become open for this ion. Decreased permeability, less and less channels remain open. So for 2 ions, extracellular positive ions, permeability increases. Actually, it goes through one and the same type of channels, because these channels are not selective and both ions can move through these channels. And when these channels are available, doesn't matter that not many of them, both ions, sodium and calcium, can enter the cell. Not many, so depolarization does not become very quick, slowly. And the third ion also participates here. Normally, a rather high resting level of permeability for potassium is not characteristic for this cell. This permeability starts to decrease and channels for potassium become gradually closed. Again, not all of them, but permeability becomes less clearly. So less potassium goes out of cell, more potassium remains in the cell. Plus the two other ions move also gradually into the cell. And as a result, it doesn't matter that each permeability individually for ion changes not much, but together all the three positive ions become gradually accumulated in the cell, and by their positive charges they progressively decrease and decrease negative charge in the inner side of membrane, which means that from minus 60, potential gradually moves to the less and less negative, which is depolarization, until reaching minus 40. And at this moment, it results in opening of many channels for sodium and calcium. So this is a slow diastolic depolarization phase. And for sinus node at rest, it goes under such an angle, approximately I would say 30 degree of angle, and it takes longer even time than the potential depolarization and repolarization phases taken together. So, and now at the threshold level, many channels open for both these ions and they go easily into cell. So this produces higher velocity of depolarization, which clearly differs from slow diastolic depolarization. Simply, depolarization occurs much faster, and at the level of potential about plus 10, the channels become closed for sodium and calcium, and K plus potassium permeability increases. And potassium, as usual, produces repolarization, because if it goes out, it takes positivity out of the cell and restores negative charge up to or down to, I would say, minus 60. But when minus 60 is reached, everything becomes repeated again. Permeability for potassium decreases again, for sodium and calcium increases again, and everything is repeated next time and next time and next. And generally, it never stops throughout the whole our life. It goes like this and goes and goes. But our heart rate is constantly changes. At rest, it changes just a little bit, little oscillations around the average value. But during changes of our state, with higher or less physical activity, it changes greatly. And these changes mostly refer to this phase that's shown in red. Exactly this slow diastolic depolarization can become faster or slower. And the other parts of the potential do not change much. They just move along the threshold level to earlier time or longer time it takes to reach the threshold. So this is main phase that is under regulation, and exactly change of this velocity of slow diastolic depolarization produces change in rate of impulses. But as you may see, each impulse, each action potential corresponds to one contraction of the heart. So frequency of action potentials corresponds to the frequency of the heart rate. And how the slow diastolic depolarization can be changed. The number of channels open, the permeability changes, can become higher or less. More channels open, less channels open, and so in this case we have faster or slower this phase of the slow diastolic depolarization. And let's see, when heart rate is changed due to autonomic nervous system influences, it's called chronotropic effect. I believe that everyone understands the meaning of the root chrono. It comes from a chronos, the time god. Chronos, and chronotropic effect refers to time, and so in this case, when we talk about heart rate, it shows that there is a change in the heart rate. And chronotropic effect can be positive, which means increases heart rate, and it can be negative with a decrease of heart rate. So, as I already said, to influence the heart rate, we need to change the velocity of the slow diastolic depolarization. Again, let's see the figure with threshold, and this is normal resting. Velocity of the slow diastolic depolarization. Here you can see how it can go without changes, but now let's make changes. And first we start with sympathetic stimulation. Sympathetic system is something that stimulates the heart. Everyone knows adrenaline as well as noradrenaline is a factor that stimulates the heart. So clearly it should increase heart rate. To increase heart rate, what we do to produce the positive chronotropic effect, we should reach threshold earlier, and next time earlier, and next time. And you see, number of impulses per each time interval becomes higher, and number of contractions therefore. And how it can be achieved? We should contribute to the same changes that occur naturally, to make slow diastolic depolarization faster. And here this contribution mostly goes through opening of more channels for calcium. Higher permeability for calcium, higher than normal, regular permeability. And more calcium ions enter the cells, and with its two positive charges, it increases the velocity of reaching the threshold. And so we can see higher number of impulses per time unit and higher heart rate. Then it's a positive chronotropic effect. We need to understand the mechanism. So again, practically everything was said, but now let it be written. It's produced by the slow diastolic depolarization, higher velocity, and it's achieved through higher entry of calcium because more channels become available. And as sodium can also pass through these channels, we can mention also sodium. But predominantly we say calcium is number one. Increased permeability for the calcium and sodium. This is the basic mechanism to increase the heart rate. But again, not everywhere, but in the heart conductive system cells, because number of contraction depends on number of impulses sent down the conductive system to the contractile typical muscle cells. So this is mechanism of chronotropic effect. And diastolic depolarization becomes faster, accelerated, threshold is reached earlier, impulse appears earlier, and excitation and contraction of cardiac muscle, typical cardiomyocytes, is reached earlier. So heart rate is increased. This is the positive chronotropic effect. Now, as for the mechanism of this chronotropic effect, we already considered partially, but let's see now the picture. And this picture should be familiar for you if you remember about the chemical signaling and mechanism of influence of noradrenaline on the beta adrenergic receptors. In the heart we have beta-1 adrenergic receptors. Let this orange piece is the molecule of norepinephrine. Beneath you can see the membrane of the heart conductive system cells. Let's consider this is a cell of the sinus node. Actually it's a membrane of this cell with extracellular fluid colored in blue and intracellular fluid colored in vanilla color. So you can see to the left side the beta adrenergic receptor, beta-1, in the heart muscle is present. And it's coupled to a G protein with its alpha subunit still combined with beta and gamma, which makes it inactive yet. And also we can see here adenylate cyclase, which is controlled by beta-1 adrenergic receptors. And also we can see inserted calcium channel closed at the moment. And negative charge inside is -60. Calcium ions concentration is much higher outside. And we can start. Norepinephrine comes and binds to beta-1 adrenergic receptors. It comes from the ends, from the postganglionic fibers of the sympathetic nerves. When it binds to, it immediately changes conformation of receptor so that alpha subunit becomes activated. It gets GTP, and becomes released from the others. So it's active G protein. And this alpha subunit binds to nearby floating adenylate cyclase. Adenylate cyclase becomes activated in turn and takes as a substrate ATP molecules and converts to the cyclic AMP molecules. Two phosphates are removed, but the molecule gets its cyclic shape, and many cyclic AMP molecules are produced. Normally when adenylate cyclase is activated, it starts to produce cyclic AMP molecules so quickly that concentration of CAMP increases in 10 in second power or third even. And cyclic AMP function is activation of the protein kinase A. For activation of one molecule of protein kinase A, it's enough to have 4 C, A, and P molecules. But they are produced in great numbers, so you may understand how many protein kinases become active. And each protein kinase can phosphorylate something. And what it phosphorylates? It phosphorylates the proteins that make walls of the calcium channels. And after such phosphorylation, the calcium channels open, additionally to the usual number of open channels. And you see, opening immediately provides additional entry of calcium. When calcium enters the cell, the conductive system cell, it's not participating in contraction greatly because the conductive system, if you remember, these cells do not have much myosin or actin. It's not their main function to produce contraction. But entering calcium with its 2+ charges produces faster depolarization, faster than before. So the values become less and less and finally reach -40, which is enough to open many channels for calcium, sodium, their entry into the cell. So let's see. This is regular with blue line, and now by sympathetic system it becomes faster. The slow diastolic depolarization becomes not as slow as before, and number of impulses increases. Here, instead of 1 impulse, we have 2. And so heart rate clearly becomes higher. It increases heart rate. So that is sympathetic system, positive chronotropic effect, and it's exerted by influence on the heart conductive system cells and predominantly pacemaker, sinus node. Then let's consider what happens in case of negative chronotropic effect, which is achieved by stimulation from vagus nerve. And again, similarly, we need to influence the slow diastolic depolarization. Just in this case, vagus nerve produces even slower depolarization, decreases velocity. Again, this is initial normal resting state. Actually, it's under vagus influence as well, but it's still regular influence, usual as always. But now we increase vagus activity higher than regular. And vagal stimulation produces its negative chronotropic effect. So that slow diastolic depolarization becomes even slower, and depolarization occurs later, and next depolarization will occur later, and so you see the rate of impulses decreases. And how it's achieved? By opposing the mechanism of slow diastolic depolarization. Let's recall that during the slow diastolic depolarization, naturally, potassium permeability becomes less. But vagus, by release of acetylcholine, produces opening of additional potassium channels. And higher permeability for potassium provides a position, slowing down the slow diastolic depolarization. And you see, the heart rate decreases because of number of impulses decreases in minute. This is a negative chronotropic effect. And intensive stimulation of vagus nerve of the heart may not simply produce slowing down of depolarization, but it can produce even hyperpolarization. Let's see. This blue line shows normal case in this picture. Sympathetic system influence shown in red. Vagus influence shown in green. And you see, it slightly decreases potential even here. But if we produce very strong stimulation, now it's shown by 2 arrows. It can produce even greater, serious hyperpolarization, so that next impulse will be generated much later. And actually it corresponds maybe to 2 contractions missed, or 1 contraction at least missed. And excessive stimulation may even produce heart standstill. It can stop heart for a certain moment. Not forever, of course. Naturally, the heart has the ability to escape the influence of the vagus, but it can stop for seconds or a few seconds and then start to contract again. So a few missed contraction can happen due to intensive stimulation of vagus nerve. So this is point which shows hyperpolarization. It's below the regular -60. It's true hyperpolarization, which delays reaching the threshold greatly. Negative chronotropic effect of the vagus is explained by higher permeability for potassium. Note, high permeability for calcium and sodium produces positive chronotropic effect because it's in the same direction which itself slow diastolic depolarization goes. But here we oppose the slow diastolic depolarization. Naturally, permeability for potassium should be decreased, and now it is increased by vagus. So, increased potassium current results in the slowing down the diastolic depolarization, later reaching the threshold, later generation of impulse, and impulse is sent later to the contractile cells, so they become excited and contracted later. So generally, the heart rate decreases. And as for the mechanism of the vagus negative chronotropic effect in the heart conductive system cells, remember, right vagus for the sinus node, left vagus for AV node, but in both it decreases it. Look, here it's simply, it doesn't require activation of the enzyme. You see, it's the case when G protein directly opens the channels. You see, molecule of acetylcholine is bound to the muscarinic cholinergic receptor. And this muscarinic receptor is also 7-TMS receptor with G protein. And here G protein, activated G protein, which is alpha subunit separated from the others, simply binds to the channel for potassium and opens it from inside. And opening of potassium channel provides loss of potassium from the cell. So while the sodium and calcium enter the cell with their positive charges, potassium takes back this positive charge outside. And so now everything depends on ratio of how much sodium and calcium enter and how much potassium goes out. With regular usual vagus high activity than resting level, potassium loss does not increase, does not predominate over sodium and calcium entry. Therefore, depolarization goes just very slowly. But in case of very intensive stimulation, we can see that potassium goes out, even taking much more positive charges out than sodium and calcium bring inside. In this case, we can observe hyperpolarization. So, decreased depolarization most often, or in very strong stimulation of vagus, it's hyperpolarization. But both produce slower heart rate, clearly slower heart rate. So this was effect of vagus on the heart rate, which is chronotropic effect. Still, we didn't consider the force of heart contraction. But let's recall again, for rate of contractions, we need to influence the conductive system cells because the rate depends on them. But force depends on the cells that have their function contraction, the typical cardiomyocytes. And so to influence the force, we need to change mostly the concentration of calcium ions in the cytoplasm of the typical cardiomyocytes. And for the influence on the force, we have the term inotropic effect. Chronotropic was for rate, and now inotropic effect. This is influence on the force of the heart contractions. And heart contractility, which involves also velocity of contraction, can become faster. It's also a component of the inotropic effect. So, to influence the heart contractility, necessary to influence the typical cardiac muscle cells and their concentration of calcium in the sarcoplasm, intracellular calcium concentration. You remember, for each muscle, higher calcium level, higher force of contraction. And let's observe, this is a positive inotropic effect of the sympathetic stimulation, which means that sympathetic system makes contractions stronger and faster. So, three important features, not only strengths. Peak tension, which means practically strength, stronger contraction, then faster contraction, and as well faster relaxation. Even relaxation is also important. The faster we do it, the earlier we can feel the heart again, and earlier we can start a new contraction. So it's all important for high activity of the heart, for high elevation of the cardiac output, which is characteristic for sympathetic stimulation. Faster relaxation means that contraction is shorter, that allows more time for refilling. But if we consider the full contraction with these three stages, if you remember latent period, then period of contraction and period of not relaxation, but restoration of initial length. So restoration of initial length, if it comes faster, it's also makes shorter the whole contraction as the full 3-stage process. So these effects mediated through the same beta-1 adrenergic receptors, but located now in the membrane of the cardiac contractile muscle cells. And again, here there's a GS stimulating protein that activates adenylate cyclase. Here I will not repeat the same scheme, just what is changed in this scheme, that the cell is not conductive system cell, but cell of the typical cardiomyocyte. Therefore, I just make the line. Look, norepinephrine binds beta-1 adrenergic receptor, then it activates GS protein, which means that alpha subunit becomes dissociated from others and moves to adenylate cyclase, binds and activates it. Adenylate cyclase activated produces cyclic AMP with great increase of the concentration of cyclic AMP molecules. Cyclic AMP, each 4 of them, activate one protein kinase A molecule, so that we have numerous active molecules of protein kinase A which actively phosphorylate proteins. But which proteins? It requires explanation, because there are few substrate proteins in the heart muscle cells to produce all these 3 important parts of the positive inotropic effects. Final mechanism. What is phosphorylated finally? First, similarly to the conductive system cells, there is a phosphorylation of the calcium channels in the outer membrane, in sarcolemma. And through these channels, we have entry of more calcium. This is the same scheme that we had for conductive system cells. And higher calcium, higher force of contraction, and plateau of action potential is maintained higher. So totally it creates higher force of contraction. But then there are other proteins that also become phosphorylated, and between them, maybe firstly we should mention specific protein. This protein is called phospholambam. Phospholambam is a specific protein which is present inside in the sarcoplasmic reticulum nearby the calcium pump. When it is not phosphorylated by sympathetic system and its positive influence, it decreases, it keeps a lower activity of calcium pump. So it's like a brake that prevents great activity of pump. It permits pump to operate in normal regular activity. But when sympathetic system becomes active and all these events occur, adenylate cyclase is activated, then cyclic AMP activates protein kinase A. Protein kinase A phosphorylates also this phospholambam. And this protein stops this inhibition of activity of calcium pump. And calcium pump becomes highly active, much higher than before. What happens due to this? During each contraction, the excitation causes entry of calcium, and a lot of calcium becomes accumulated in cytoplasm and participates in contraction and provides high force of contraction. But you remember that heart should all the time after contractions become relaxed, so that feeling may occur, and only with feeling next contraction will be useful. So to make relaxation, this calcium should again be decreased back to normal resting concentration, and calcium pump is the main structure that provides this decrease. And notice that calcium pump is placed in the sarcoplasmic reticulum. Calcium enters to the cell from 2 sides, from outside and from reticulum, and even less from reticulum than from outside. But now, when sympathetic system becomes active, calcium is pumped back mostly to the reticulum. So reticulum receives not only calcium that is released from it, but also part of calcium that entered from outside. And therefore concentration of calcium in the reticulum increases with each contraction. And the highest concentration, the more calcium will be released in the next opening of channels. Therefore, with each progressive contraction with high activity of pump, we have greater release of calcium. Of course, it's not immediate effect, it requires repetitive contractions. But due to the greater uptake by sarcoplasmic reticulum of this calcium and greater storage, greater release subsequently will occur. And immediate effect is faster relaxation. The faster calcium is decreased back to normal resting level, the faster the cardiac muscle will become relaxed. So it can be filled with blood faster, and we can start new contraction earlier. So actually this mechanism provides also the basis for a higher heart rate even. But generally we consider it as a part of a positive inotropic effect. Greater activity of the pump, which is provided by phosphorylation of the protein called phospholambam. So we see two consequences. First, easiest, the faster we decrease calcium, the faster relaxation occurs. And then calcium is taken not everywhere, not outside, but mostly into reticulum. And reticulum can store more and more and more progressively calcium. And the release can be increased later. As for the negative inotropic effect of the vagal stimulation on the heart, first we can recall that vagus fibers innervate in the cardiac muscle part, not conductive system, only atria. And this effect is exerted mainly in the atria. Acetylcholine binds to the muscarinic receptors. Then it activates G inhibitory protein, GI, which inhibits adenylate cyclase. It's exactly opposite to the sympathetic stimulation by GS protein of adenylate cyclase. And so, opposite effect to activation of beta-1 adrenergic receptors produces decrease in contractility. Look, cardiac muscle cells, potential for atria. Naturally, it should have prolonged plateau phase. When calcium comes inside, potassium goes outside, and these two currents are balanced in charges so that the potential remains over zero, positivity is kept, and still calcium is present and contributes to contraction. And only when calcium channels become closed, potassium easily removes positivity from cell and restores resting stable potential minus 90. Now let's see what happens. The phases numbers are here again to remind you that it starts not from 1 but from 0. But look, acetylcholine decreases current of calcium inside because of opposition to the adenylate cyclase activity to beta-1 adrenergic receptors. And also potassium increases outward potassium current, because similarly to action in the conductive system cells, it also increases potassium permeability. And look, if calcium entry decreases, you see arrow becomes smaller, and potassium going out increases, there will be not long plateau phase. Plateau phase becomes shorter and polarization occurs earlier. And amount of calcium that enters during the plateau phase is less, and exactly this calcium contributes to high force of contraction. And now with less calcium, force of contraction is less, and potential becomes shorter, really. So this is influence on the heart of vagus. And doesn't matter that ventricles do not have vagus effects. Finally, ventricles also become contracted with less frequency and with less force. So decrease in tension for the heart contraction. As for two other components of two other factors for the heart, main factors, heart rate and heart force of contraction or tension, and also there is two factors. There are conductivity and excitability. These effects also have their names, and it's not easy to memorize, but I hope that you are able to do it. For the conductivity, this is dromotropic effect, dromo. And for excitability, bathmotropic effect, or bathmotropic. So these both effects, again, for sympathetic system are positive because it increases conductivity, so conduction goes faster between cells, as for conductive system cells in the conductive system, and as for typical cells, they also conduct faster. In excitability also becomes higher. As for parasympathetic nerve, here we have both negative effects. there is a decrease of conductivity, decrease of excitability, which finally really contributes to lower tension of contraction even for ventricles which are not innervated by vagus. But conductive system, conductivity decreases, excitability decreases, and so we have this effect easily of less force of contraction. So again, these two effects of conductivity and excitability, dromotropic and bathymotropic, contribute to the main effects, actually, to the influence on the force and on the rate of contractions. And fortunately, it's easy to memorize. All four effects are positive for sympathetic system, and all four are negative for parasympathetic vagus. Then we move to cardiac output, which is stroke volume multiplied by heart rate. Neural mechanisms. Here, first we consider just background for neural mechanisms. The efferent part of reflex arches. You see, before now we considered only efferent mechanisms, how efferently from sympathetic nuclei or from vagus nuclei we can change heart activity. But the heart should be stimulated, when it should be stimulated, when it should be suppressed. It depends on situation and how to determine this situation. Everything should be arranged in automatic mechanisms of regulation. And now, background for all these mechanisms, how they operate. First, as we considered efferent mechanism, now it's necessary to make the full reflex arches, which start from receptors, then go to centers, and from centers, efferent mechanism already become switched on and regulate heart activity. So, but basics mean that it's arranged on the negative feedback principle. So simply, when cardiac output increases or decreases, we should do something to make it opposite. In case of increase, we should decrease back. In case of decrease, we should increase it back. In a steady resting state, we maintain the cardiac output, which is product of stroke volume multiplication by heart rate. And in this case, maintaining this cardiac output at a steady level requires to decrease both rate and tension, or in case of cardiac output elevation. If cardiac output becomes too high for resting state, we need to decrease both rate and tension, because tension determines the stroke volume. And then cardiac output comes back to normal. Or we need to increase heart rate and tension of the contraction in the opposite case, when cardiac output decreases and we need to restore it back to normal, make it higher. We need to increase both stroke volume, which is determined by force of contraction of the heart, and heart rate. This is clear, easy, negative feedback. So tension is force and heart rate is heart rate. So both inotropic and chronotropic effects are necessary to regulate the cardiac output finally. So, and cardiac output brings necessary amount of blood per minute for organs and systems, depending on their requirement. So let's consider efferent parts for regulation system. These efferent parts are presented by baroreceptors and chemoreceptors. For the baroreceptors, they are mechanical and respond to pressure of how much blood compresses the vessels, and especially the areas with great amount of receptors. Greatest amount of receptors is located in two areas, carotid sinus and aortic arch. Baroreceptors respond to pressure and stretching, which actually are related to each other. Higher pressure stretches arterial wall stronger. So pressure and stretching go in parallel, usually. So higher blood pressure, the higher is rate of impulses from receptors. And blood pressure becomes higher when stroke volume and heart rate increase, both or each individual. Impulses from receptors are conducted to the brainstem. From carotid sinus, they are carried by the fibers of the carotid sinus nerve, that is a branch of glossopharyngeal nerve, number 9. And fibers from aortic arch are fibers of the vagus itself. Vagus is the complex nerve. It contains the fibers that go from central nervous system to periphery, like innervation of the heart, but also it contains fibers that go from receptors, from periphery upward to the brain. So exactly these sensory fibers are present in vagus and come from aortic arch directly to the nuclei of vagus nerve. So look again at the placement of these areas, which are called reflexogenic. They produce reflexes, these reflexogenic zones, receptor zones. First, this is aortic arch. Why here we have one of main detectors of blood pressure? Because this is entry to the whole systemic circulation. So we control pressure in the beginning. Aorta is the beginning of the systemic circulation. But also we have separate control. Here, by the way, this is nerve vagus itself carries impulses from aortic arch receptors to brain. And also we have two other areas, two carotid sinuses. Sinus is the dilated portion of carotid artery just before branching of this artery and division into internal and external carotid arteries. And additionally, we constantly monitor blood pressure, and not only, but blood composition, content of oxygen here, because this is entry to the blood supply to brain. It's important. Even if the whole body, rest part of whole body, will have good blood supply and brain will not, this will result in bad situation for the body as a whole. So we have to monitor not only general blood pressure and blood quality that supplies the whole systemic circulation, but also separately brain, because without brain the whole body will die. So entry to brain blood supply is monitored, and here it's important reflexogenic zone, pair of them. And nerve number 9 goes again to medulla and carries impulses. But finally, from medulla centers of the 9th nerve, the impulses will go to necessary centers. And centers we need to consider now. Brain stem centers for the heart regulation. All are located in medulla. Reticular formation, medulla, and a little bit in the lower 1/3 of the pons, but predominantly the medulla. And it's all within reticular formation because it's called reticular because of numerous net-like connection between neurons. Because so many functions are associated with each other, so many connections are required between neurons that control vital functions. You know, these centers belong to the vital centers or essential for life centers. These centers receive information about blood pressure from baroreceptors, and then they may change output to the heart through parasympathetic nerves or sympathetic nerves of the heart to correct the blood pressure, and it's organized in a negative feedback principle. Efferent carotid sinus and aortic nerves stimulate the activity of vagus nuclei and increase the vagal effects on the heart, decrease of force and rate of contractions finally. So you see, first, baroreceptors respond to the blood pressure proportionally. The higher becomes blood pressure, the higher is the rate of impulses generated by these receptors and sent by sensory nerves from carotid sinus and aortic. And so when the receptors respond by higher activity to higher pressure, they clearly should stimulate something that directed to opposite changes. And exactly they stimulate nuclei of nervus vagus, this portion which controls exactly the heart activity and controls by negative effects, negative chronotropic and negative inotropic effect. Not to say about the others, batmotropic and dromotropic, all negative effect. So let's try to sum it up by formation of a simple scheme of the negative feedback regulation system. Let it be medulla, this portion. And exactly within medulla we have nuclei of the nervus vagus, which take large portion of medulla. And somewhere, not far, we have the heart. For schematic purposes, we'll substitute by simplest symbol of the heart. And you see, heart produces certain level of blood pressure, which is controlled in two areas. Blood pressure controlled by aortic arch baroreceptors and by carotid sinus baroreceptors. So higher cardiac output contributes to higher blood pressure, and to higher activity of these receptors. These receptors in turn send their impulses by aortic nerve, which is part of vagus, and sinocarotid nerve, which is a part of nerve number 9. All these impulses come to medulla and finally activate not directly, but finally activate the nervous vagus nuclei, the portion, the number of neurons that directly control the heart. And so finally vagus fibers send impulses to the heart, and these impulses produce negative effect, decrease of heart rate and tension. Under normal conditions, in resting state, all this system is well balanced, and vagus produces its constant negative influence to the heart, but resulting heart rate and resulting tension is normal for us, so that we have classical heart rate between 60 and 90, so about 75, and we have regular blood pressure and tension developed, so blood pressure is maintained on a regular level. Therefore, both receptors of aortic arch and carotid sinus, all these baroreceptors, have their regular, usual rate of impulses, which stimulates nervous vagus nuclei with usual frequency. And it maintains activity of nervus vagus on its regular level, so that vagus constantly sends impulses one after one to the heart and keeps it with its normal heart rate and normal cardiac output. This system is balanced. But in case of higher than necessary activity of the heart, all these influences increase and stimulate vagus so that it suppresses the heart more than usual. And we have this decrease of heart rate and decrease of cardiac output more to values less than regular. And if heart rate and tension decreases due to different reasons, cardiac output is decreased and blood pressure decreases. In this case, activity of baroreceptors decreases proportionally. And much less activity of baroreceptors, much less activate nuclei of nervus vagus. In this case, vagus influence also decreases proportionally, and heart gets less inhibitory impulses. So heart rate and force increases. So presence of resting activity of vagus nuclei on the heart we can observe if we block the heart. In experiment in animals, the nerves can be simply cut, but also it can be blocked by pharmacological pharmacological influences like muscarinic receptors block or so. If this blocked, the heart immediately gets less inhibition, and less inhibition produces much stronger contractions of the heart, and heart rate becomes higher. So absence of constant, regular negative effects of the vagus will immediately appear as stronger heart contractions. I hope it's clear, but usually we are under constant control from vagus when we are at rest. Just imagine that something important for you, think about something important for you, immediately vagus influence decreases and sympathetic system influence increases. But at rest we have vagus control. And many reflexes are arranged through vagus nerve. And actually it's very easy to predict, if you know about any reflex, that this is vagal reflex. Actually you may say that anyway final effect becomes decrease of heart rate and force of contraction if it's arranged through efferent fibers of vagus. Examples. First, Ashner reflex, what is also it's called ocular cardiac reflex. If we press the eyeballs, of course without very strong force, moderate press in the closed eyes through lids produces decrease of heart rate. The mechanism of this reflex goes from receptors in the eye, and these impulses from mechanical receptors are sent through the oculomotor nerve to the medulla, and then it goes to vagus nerve nuclei, and from vagus nerve nuclei down the vagus nerves, peripheral nerves, it goes to the heart and decreases the heart rate. It can be rather useful for people with sudden attacks of tachycardia. If it happens more or less often, the people can be trained to use this reflex to prevent serious tachycardia, because in the beginning of attack it's possible to suppress it by activation of the vagal reflex, such as Ashner reflex. So if patient feels that tachycardia just starts to begin, starts to be produced, so the pressing of the eyeballs can suppress this and attack can be overcome. Not necessarily always, of course. Then Gold's reflex. Such a reflex can happen, for example, during boxing in the ring. With very strong punch to abdominal area, there is a stimulation of the solar plexus, which contains vagus fibers sensory, and great stimulation with sensory fibers comes to the vagus nuclei, increases activity of vagus nuclei neurons, and as a result, vagus nerve produces in response high influence on the heart back, and heart rate decreases, and even it can produce stop of the heart for a short time. For a few seconds it can be stopped to heart standstill. Then usually the heart starts to work again, but at least this moment it greatly decreases the heart rate or makes a missed few contractions. Gold's reflex. Up to reflectory cardiac arrest. Cardiac arrest means the stop of the heart, standstill. Then, simply according to previously seen schemes, you can see that fast increase, sharp, immediate increase in the systemic blood pressure produces decrease of heart rate and contractility, which it goes simply through baroreceptors. Systemic blood pressure increases, aortic and sinocarotid and carotid sinus receptors increase the activity correspondingly. They stimulate vagus nuclei in medulla more, and vagus produces stronger inhibition of the heart. Not only systemic blood pressure, actually similar mechanisms exist in the pulmonary artery and in the pulmonary circulation. When blood pressure suddenly starts to increase in the pulmonary artery, this produces decrease of heart rate and contractility similarly to the changes in the systemic circulation. So these all reflexes arranged through vagus nerve. Now, let's sum it up again. The whole autonomic heart regulation reflex arches. First, aortic arch and carotid sinus artery and carotid artery have their receptors, and sensory fibers go to the through 9th nerve, and 10th vagus itself goes to the medulla. And this is shown in red now. It's medulla portion, not spinal cord. Don't mix it. And it binds to, it stimulates certain neurons that finally activate vagus nuclei. And vagus nerve goes down to the heart and sends more impulses than usual to the heart conductive system and atria, sinoatrial and atrioventricular nodes. So the heart activity decreases. And as for reflex arches for sympathetic system, it arranged through thoracic segments, upper 3, 1, 2, 3, and through stellate sympathetic chain, stellate ganglion, and sympathetic cardiac nerves finally come to the conductive system, atria, as well as ventricles. And ventricles, I hope you remember, make difference from vagus fibers. Also, we can see here that impulses can be sent down to centers in medulla from hypothalamus. The hypothalamus is higher autonomic center, as you remember. And changes of activity in hypothalamic neurons in anterior group of nuclei and posterior group of nuclei can easily make shifts of balance between vagus influences on the heart and low influence for resting state from sympathetic system. When activity starts, physical activity or emotional stress, we have high activation of the posterior hypothalamus group of nuclei, and then it decreases activity of vagus and increases activity of the sympathetic system, so that heart functioning becomes at higher level and cardiac output increases, blood pressure increases, heart rate, and so on. Chronotropic, positive, inotropic positive effects, and so on. All that we considered by now was neural mechanisms. Actually, they take much more time to explain and understand. As for extracardial humoral mechanisms of regulation, now it's rather easy because we know receptors in the cells which can respond to hormones such as adrenaline, noradrenaline, epinephrine, norepinephrine, catecholamines. When doesn't matter, they are released only as transmitters from nerve endings of sympathetic nerves, or they come through blood from the adrenal medulla. The final mechanism is the same. Difference is just how quickly effect occurs and how long it lasts. But the mechanism is the same through the same beta-1 adrenergic receptors in the heart muscle cells and in conductive system cells. And therefore all positive effects develop in response. So influence of catecholamines is clear. I hope everyone may understand how it's arranged. Then angiotensin II, this substance we'll repeatedly consider more and more. This substance generally, according to name, angion, vessel, and tension, it's clear, will produce vasoconstriction. It's a powerful vasoconstricting substance, but as well it has its influence on the heart and predominantly increases heart rate, but also contractility. Then thyroid hormones. I hope you remember that thyroid hormones may produce their permissive effect for catecholamines, and they increase sensitivity of beta-1 adrenergic receptors, so that you perhaps remember that excessive level of the thyroid hormones may produce tachycardia and generally stronger contractions and higher cardiac output for the heart. So it's because of higher sensitivity of beta-1 adrenergic receptors in the presence of the thyroid hormones. Normal level of hormones just contributes to normal reaction of these receptors, but abnormally high or low level correspondingly will greatly increase or decrease receptors sensitivity. And perhaps you remember that also the other group of hormones have similar permissive effect, glucocorticoids. And they also increase heart rate and contractility because they generally, all beta-adrenergic receptors, sensitivity, they increases it. And so, again, with excess of glucocorticoids, we can expect higher heart rate and higher blood pressure due to higher contractility of the heart. This is both last two hormones groups, thyroid and glucocorticoids, produce their stimulating effect on the heart by higher sensitivity of the beta-adrenergic receptors to the norepinephrine, epinephrine. These are permissive effects in both cases. And the last thing I'd like to consider, this is effect of neurotransmitters and ions on the heart, and how we can explain it. Actually, the effect of neurotransmitters should be very clear by this moment. But as for ions, they also have effect. These effects we are going to observe on the record from the isolated frog's heart, like we can do from mechanocardiogram. It's mechanical record when ascending part shows contraction, systolic, then relaxation and diastole. So each loop that goes upward shows one mechanical contraction of the heart. So this is normal free contraction, example, initial. Then we add acetylcholine. Actually, it should not be something difficult to predict because acetylcholine is the transmitter of nervus vagus. And all the effects of vagus should be really achieved now, both negative chronotropic and negative inotropic effect. So we should observe decreased amplitude of the contractions, which is proportional to tension, to force developed, and decreased rate of these contractions. And observe, addition of acetylcholine decreases progressively both force and rate of contractions. Then, let's observe what happens in case of great elevation of the potassium level, excess of potassium ions, hyperkalemia. Actually, if you recall that acetylcholine produces its influence by higher permeability for potassium, you can observe the common features of these two effects. Excess of potassium also produces decrease in heart rate and in heart force of contractions. Look, but if this excess is great enough, potassium excess, It may not simply decrease heart rate and force of contraction, but produce a hard stop. Heart relaxes and stops, stopping diastole. Actually, it's even used in the medical practice. Solutions with great level of potassium, so-called cardioplegic solution, that produces paralysis of the heart, and they are used for heart intended for transplantation, to stop it and not operating until it's necessary. For transportation, it should not beat like it's shown in some movies. They beat inside the boxes, but potassium excess produces stop, standstill of the heart, and it should, of course, the cooling down is necessary to keep it at low temperature. But excess of potassium can produce this stop in diastole. At least for frog's heart, it can be observed. And it's cardiac arrest. Termination of contractions, it means that blood movement is stopped at the moment and blood circulation is not maintained. So now the opposite case, adrenaline, epinephrine. Also, it's predictable what happens in case of release of adrenaline. All positive effects of the sympathetic system. We add, and we can observe high force, which appears here as the amplitude, and high rate of contractions. So it goes up until certain limit when it reaches the reasonable limit. So, and now calcium ion excess. Not simply calcium, but excess of calcium ions also contributes to higher force and higher rate of contractions. Let's see what happens. There is similarity, but not entirely. Look what happens. Finally, imagine heart contracts, relaxes. Heart contracts, relaxes faster and stronger. But then heart doesn't reach full relaxation during diastolic relaxation and contracts stronger. Then next time again, relaxation is not reached, and gradually it becomes contracted stronger, stronger, and finally stops. And here it's stopping the systole. But I hope you understand actually that it's not great difference for the blood circulation. Heart is stopped in the diastole or in systole. In both cases, this is cardiac arrest, and blood circulation is not maintained anymore. So in both cases, it's a death for a body actually, which comes very soon. And why does such difference exist? You remember that adrenaline operates mainly through opening of more and more channels for calcium, and calcium excess can provide similar effect. But there is big difference. Adrenaline produces full relaxation, while calcium excess does not. And how it can be explained? If you recall the mechanism of adrenaline inotropic effect, you may remember that it does not only contribute to higher entry of calcium during systole, but also it stimulates faster pumping of calcium back to reticulum during diastole, during relaxation. This effect achieved by phosphorylation of the protein phospholambam. This is important. Heart not only faster and stronger contracts, but easily and faster relaxes. This is action of adrenaline, which is multi-directional. As for calcium excess, it doesn't provide any conditions for faster pumping of calcium. It enters, but pumping is not stimulated. Without sympathetic stimulation, pumping remains as before, and calcium excess develops very quickly, easily, and calcium becomes simply greatly accumulated inside the cytoplasm, which contributes to greater and greater degree of contractions. And finally, heart, especially in experiment, of course, in the real body it's hardly achieved, but in experiment in isolated frog's heart, we may observe that heart contracts maximally and stops in this position for a certain time. So this was the influence of the neurotransmitters and comparison of ions, basic ions, potassium and calcium on the heart, shown on mechanocardiograms of the isolated frog's heart. And with this, I hope we can finish. Thank you for attention, and I wish you successful studying.